strong.
Hi everyone! You are now watching Biomix Daily Top 3. I am VJ Pearl.
And this is VJ Aya. Happy Friday! Welcome to the show where we feature the hottest and the coolest educational music video in town.
So guys, are you ready na ba to know our new entry of this week? Kasi ako ready na. But before that, did you know guys that in 1952, Dr. Edwin Chargaff discovered that the amount of thymine and the amount of adenine was roughly the same as whereas the amount of guanine and cytosine. And this discovery of Dr. Edwin Chargaff later became known as the Chargaff rule. So guys, enough with that trivia anymore and let's proceed to the countdown. At number 3, protein synthesis by Mr. Rock.
Look at what's coming out of the nucleus, destined for a ribosomal strand of mRNA. It's got the code, the information for the protein we'll be making today. The RNA message is organized into codons. That's a sequence of bases 3 that spell out one and only one amino acid, a genetic code that works universally. A codon has its match in an anticodon, 3 bases on tRNA's bottom side. tRNA's job is to bring amino acids to ribosomes so proteins can be synthesized. mRNA, tRNA, and ribosomes plus amino acids make a protein making machine. Read the codon, make a polypeptide, that's translation. Cells synthesize proteins. The small subunit of a ribosome binds with an mRNA's leading end and slides until it reaches start codon AUG, indicating where translation begins. A ribosome has 3 binding sites where tRNAs can bind. The A site's first piece, next then E, and the E's got an exit sign. The first tRNA in this translation process binds a start codon and binding site P, and that tRNA carries that amino acid that goes by the name of methionine. And now, to finish this initiation, the ribosome needs to be made complete. The large subunit binds a small subunit. Now we're ready for translation to proceed. mRNA, tRNA, and ribosomes plus amino acids make a protein making machine. Read the codon, make a polypeptide, that's translation. Cells synthesize proteins. Now we're ready for the next step, elongation. An amino acid carried by a new tRNA with an anticodon matching with the second codon binds at the site called A. Then the ribosome carries out an enzymatic action binding these amino acids with a peptide bond. So a dipeptide hangs on the A site tRNA. Watch translation move along. Next, the ribosome shifts exactly 1 codon over in a move that we call translocation. So the A site's empty and the P site's loaded, and the first tRNA's in the E location. E site tRNA makes its exit, and the next tRNA fills a site called A, and the ribosome catalyzes away. In your cells, it happens every second, every day. And watch that polypeptide, see it get longer as each codon gets translated. Ribosomal robots read RNA instructions as proteins get created. mRNA, tRNA, and ribosomes plus amino acids make a protein making machine. Read the codon, make a polypeptide, that's translation. Cells synthesize proteins. Protein synthesis, translation. Protein synthesis, translation. Protein synthesis, translation. Protein synthesis, translation. We started with initiation, which was followed by part 2 of translation, elongation. Now it's time to finish synthesizing this protein with a finale called termination. When the ribosome gets to a stop codon, there's no tRNA anticodon that matches, so A site's bound by a release factor, the polypeptide detaches. tRNAs of P and E sites float away, ribosome breaks apart, no longer one, the polypeptide folds into a functional protein. Translation is done. mRNA, tRNA, and ribosomes plus amino acids make a protein making machine. Read the code, now make a polypeptide. Now translation, cells synthesize proteins. mRNA, tRNA, and ribosomes plus amino acids make a protein making machine. Read the code, now make a polypeptide. Now translation, cells synthesize proteins. Protein synthesis. Translation, 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 protein synthesis.
And that was Protein Synthesis by Mr. Rock.
Greenish Take Home Bestsellers! Pasensya, istorbo.
Trabaho lang po.
Greenish Take Home!
Ay, nakita na niyo to.
Sorry.
Ay, baliktad.
Greenish Take Home Bestsellers! Uy! Greenish Take Home Bestsellers! Sorry po.
You know, Hex
Greenish Take Home!
Oo naman, Pearl, that song was really, really groovy. But before anything else, may patrivia rin ako eh. Kilala niyo ba si Rosalind? Hindi si Ate Rosalind na nasa gate, guys. Yes, it's Rosalind Franklin. So Rosalind Franklin was an English chemist and an X-ray crystallographer that studies the molecular structure of DNA and RNA. So without further ado, let's now listen to the DNA song by Dan Campos, which tops second on our playlist.
Deoxyribonucleic acid contains your genetic code. DNA, a molecule that makes up all your chromosomes. Double helix structure, a two-stranded spiral. Set of instructions for your cells so that they know what to do, like send their signals and make new bonds. Making proteins from acid to amino. Complete set of genetic instructions is a genome. Nucleotides like genes and have to think that they're composed of phosphate, nitrogen base, and deoxyribose. Each nucleotide base is made up from these four: adenine, guanine, thymine, adenosine, and that's all. A always pair with D, and that's the base pair roll. C always pair with G, and that's the way they go. Deoxyribonucleic. DNA is deoxyribonucleic acid, yeah. Deoxyribonucleic. DNA is deoxyribonucleic acid. These complementary bases, each on opposite strands. You can remember the pairs, both the mnemonic GCAT. Telecontrol complex process to make a protein from a gene. It goes transcription and translation to intercepts in the journey. Transcription takes information, transfers to mRNA. Messenger moves the nucleus and onto the next stage. This is called translation. Happens in the cytoplasm. Ribosomes read the code on tRNA. Makes proteins one as amino at a time. Deoxyribonucleic. DNA is deoxyribonucleic acid, yeah. Deoxyribonucleic. DNA is deoxyribonucleic acid, yeah.
Now, first on our playlist, I bet you know these people, Pearl. They are our classmates back in college. Naman, these people are very, very smart. Very, very talented pa. Indeed, favorite talaga ni Lord.
So enough with the trivia, Aya. At number 1, Protein Work by Jay Sanglitan at Sofia Bisilia.
cause my cells are working. They're synthesizing the proteins cause of central dogma. There are 3 stages that you really gotta know. There is transcription, editing, and then translation. I know you're always crying the night before, and you can understand all the lessons. But we are here right now to help you, so you can get that perfect scores. First you gotta learn the code, and it always says man, and we go for 3, 3, 3, 3, 3, 3, 3, and we finish at the stop. We start before a polypeptide. There are 20 amino acids. We got initiation, and then elongation, and finally in transcription we got termination. Can you guess where this happens? At the nucleus. And don't forget tatabox and RNA polymerase. And then we end up with pre-mRNA, which undergoes some editing, where there's a 5-carbon polate tail, and they help with degradation. Now you know we gotta splice, with the help of slurps, and you just gotta be cut out. Well, the exits gotta stay. Then you can get diversity from alternative splicing.
Ooh yeah, translation. In the cytoplasm, we use the RNA to bring amino acids together. The small subunits bind to the RNA, and it moves down the stream until the start codon. Whoa! Then the large subunit attaches to the strand, completing translation, initiation, complex, and now tRNA can get to work.
Agape side. Next is A side. Except for the E side. And RNA is broken down. The polypeptide that is released, and that is how you synthesize. Protein synthesis. Protein synthesis.
So we hope you enjoyed the hottest entries of this week.
So, see you again next week, guys. Bye!
Bye!